Can everybody see it?
Yes.
Yes.
Thank you for your confirmation. Good morning, everyone. Today I'm going to talk about machine learning techniques to predict the effectiveness of music therapy. In this paper, I will introduce a study to set up the predictive factor of music listening relaxation effects using machine learning methods. As you know, music listening is a universal technique used by music therapists in several clinic settings, whose mainly purpose is psychophysical relaxation. In this way, machine learning techniques were used to investigate how personal factors such as age, sex education, musical training, musical listening frequency can predict the effectiveness of two music listening approaches. Individualized and melomix health musics. Regarding materials and methods, more than 300 participants were enrolled and designated to two groups divided by sex, age, and education. In this slide, we can see that both table 1 and table 2 provides information about the actual age distribution of the participants, as well as the distributions of education levels, music and training, and sex. With this in mind, decision 3 were used to predict the effects of music listening on relaxation, as you see here in figure 1. 320 participants were selected without health problems, and they were distributed by age and education level, presence of musical training, and sex, as we mentioned previously. After randomization, each of them listening to music for 9 minutes, for 9 musics, either to their preferred musics, IMLG, or to algorithmically generated music, MHG. As we mentioned in the last paper, decision trees are selected because they are simple to visualize, and they allow us to evaluate the importance of each feature in the final results. Now, we are going to talk about the results. Here in table C, we can see relaxation level. We recorded using a visual analog scale with a range of 0 to 10 before and after the listening experience in different music listening conditions. Melodic health, individualized listenings. At the same time, participants were divided into 3 classes: increase, decrease, or not change in relaxation. So, on the other hand, in figure 2 shows the case in which participants listening to melodic health music, and each leaf node is labeled with the classification choice. White equal positive effects, black negative effects, gray no effects. The first 2 splits are performed by relaxation level and age, and the next factor appearing is the education level, while figure 3 shows a less complex decision tree, in which the first splits are on the relaxation levels and the listening frequency, followed by musical genre, by the level of education. The result of the test sets for the decision 3 for figure 2 and figure 3 are shown in this table phi, where the F1 score is defined as the average of the precision and recall. It is used as a statistical measure to rate performance. As a final conclusion, we can say that this study show which factors can influence the relaxation effects, or individualized or algorithmic music listening via machine learning techniques can potentially be considered an important methodological approach in this field of music therapy. That's all. Thank you for your attention.
Okay, I'm going to share my screen. Okay, now, I'm going to talk a little bit about this article called "An Inexpensive Microscopy System for Microfluidic Studies in Body Yeasts." So these scientists develop a minimal cost hardware attachments to provide long-term focus, stabilization for lower cost microscopes, and open source software to manage current time-lapse image acquisition for multiple microscopes. Uh, in order to know what is the microdissection assay that they use in this study, they know that is a bottleneck that limits progress of aging research in the body yeasts. So to measure the lifespan, it is necessary to acquire a multi-day time-lapse comprised of high-resolution bright field images of yeast mother cells, about 3 to 10 micrometers in diameter, aging in a microfluidic device. So I think with this is a very interesting study because we are now working with yeast, and we have a little bit problems with this. So the central challenge in long-term microscopy imaging experiments is maintaining the sample in focus against vertical stage drifting and caused by thermal fluctuations and mechanical slag in the focus system, or other source of tension. This automated compensation is possible if the distance between the objective and the sample can be adjusted by motorized microscope components. So, what about the design? To create an economical solution that could be easily adopted by a wide variety of laboratories with minimal engineering expertise. They choose to balance the convenience, cost, and performance of these extremes, but by starting with budget-friendly upright compound microscope. They choose stand and optical components that provide high quality images, and design the necessary components to motorize the axis of the stage. On the software side, they create a graphical user interface based software to control the motorized components, drive a robust image analysis based autofocusing system, and manage the current collection of time-lapse images for multiple microscopes. In the results for microfluidics experiments, they were serially diluted and growed overnight at 30 degrees. The microfluidic device were filled with 2% of glucose medium. The cells were loaded into a 5 milliliter syringe and injected into the device by hand. And the media was flowed through the device via syringe pump at a rate of 2 to and 10 milliliters per minute. And the cells were imaged every 5 minutes for 120 hours. And in conclusion, the instructions for the use and assembly, the CAD designs, files for 3D printed parts, and executable software files as well as source code are available by download for the K Berlin Lab. That's it. Thank you.
Good morning, everybody. I present this research called lung cancer detection using CT scan images. So in this case, lung cancer is aborted because this is a mortal disease. The diagnosis it's very difficult for doctors. So in this case, the research is focused in early detection according to the information from CT or computer tomography images. So this method abords different kinds of machine learning algorithms in order to improve the literature review. For example, in several methods proposed, the models are focused in the segmentation images and interpreted this information according to accuracy measure, sensitivity, and specificity. So different kind of machine learning algorithms are used. For example, convolutional networks, fuzzy algorithms, and image processing methods with machine learning. So the stage of the methods are described in this image. For example, the CT scan images are input to the system. In this stage, the image processing is performed. In this case, some filters can be used in order to improve the information from images. The next stage is according to the segmentation method. And finally, and the main procedure of this model is the feature extraction. In this case, for example, the feature extraction is according to the perimeter or area, the information from the images. And finally, and this the improve of the proposed method is the classification method using machine learning. In this figure is presented the stage of the proposed method where the several stages are added in order to improve the results. For example, the new stage is using the image preprocessing using other filters, and the stage of feature extraction with different characteristics. For example, area, eccentricity, diameter, centroid of the regions of interest identified in different SETI image. And finally, and the improved stage also is focused to use support vector machine in order to train a model from prediction. In this case, the nodules identified in images can be classified or with benign or malignant nodule. The methods are evaluated with 1,018 cases from a real data set medical patient. Normally, the images in medical files are in DICOM format, but the computer system in general can be used in GPG files or PNG extension from images, which are converted. And super vector machines are trained with a free database.
1 minute left.
LDC. Okay. So this figure, the process of processing and training the method are presented were original image, grayscale, and finally the image with the region of interest detected. So in this case, for example, two nodules are detected with different perimeters and area, which is the information to input the supervector machine. The accuracy obtained with the proposed method is upper 90%, which is very evaluated in medical file, and the sensitivity is upper of 50%. The time of training, that is another important parameter in the computer system, are in order for 5 seconds. The accuracy is evaluated with a positive true and negative true evaluations, and well, so these graphics present the data for training the method of machine learning. Finally, the classification of nodules are presented in this table, where in different images, the nodules can be classification for true or false in parameter of maligned. So finally, the method of machine learning can be used in medical file in order to improve the diagnosis of different disease, in this case for the lung cancer, with highest accuracy. Thank you.
Can you see the screen?
Yes.
Okay. My paper is the ultra sensitive part 1, Michelian response and zero order ultra sensitivity. Sensitivity. Okay. This paper is the most important to show you that the relationship or the initial response, which is a mathematical models that describe the cooperativity behavior in cellular systems, and the experimental system which are described with that models. The sigmoidal shape is the plot more frequently appear in biological systems. The sigmoidal curve is that is shown in these figures, and this curve means that the or input signal in order to 20 to 80%, and the response of the system are more or 80% the response. In this case, is the sigmoidal curve the best plot for describe this behavior. Many examples the biological system are described with that dynamic. In the table 1 is some example, but in this exposition, I speak only one system, the phosphorylation desphosphorylation monocycles. In figures 2, the Hill function are the theoretical model for describe the sigmoidal behavior. Output in this equation with n is the integer number. Integer is 1, 2, 3, 4, 5. And if you change the exponent, then the figures is evolution to the sigmoidal behavior. In the another figures, the relationship in between output and input signal, this relationship is defined with local sensitivity, and local sensitivity is function of the exponent of the sigmoidal plot. In books, in this equation is the Goldberger-Koshland equation, and this can describe the sigmoidal behavior. Sigmoidal behavior very well. And one example of this efficient description I show you in these figures. Figure
1 minute
5.
left.
Here I zero order ultrasensitivity in the monocycle, the phosphorylase and phosphorylation systems. The point are the experimental data, and the lines red or blue correspond to the models, to the mathematical models. We can see that model is very well for lowest concentration of substrate. Substrate is the input signal, and output signal is the relationship in between phosphorylase and total phosphorylase. In conclusion, this mathematical model, this equation, can be used in ground cellular. In ground cellular, I believe that model can be used and see the relationship between substrate or input, which in this case correspond to the media culture and number of cells or ground velocity, the output in these systems. I believe that can be very interesting study that system with the Goldberg's Cosland equation. Thank you very much.
Okay. My topic is high-speed large-scale for the activities mapping on moving C elegance by deep learning enable live file microscopy on a chip. This is a new fusion microfluidics and light file microscopy. Is disclosed to achieve high speed for the image of C elegance in motion inside the chip. Thanks to this, it is possible to quantitative analyze behavior as well as neuronal activities and correlate phenotypes with the function of neurons. In this part, platform is created for image capture, automatic analysis of high performance C elegance. A value-based chip was designed to manipulate C elegance by adapting the size of the worms. And LFM is sequential live file. Its system was specifically designed to record the movements of C elegans, and several worms were loaded into the reservoir and drive on the image for 1 minute. Um, in this part, deep neural network is used to transform route to the light file image into a 3D volume sequence with notably better quality than the live file method. Um, well, well, the chip works with a lot of words, capture, download. These are displayed in the LabVIEW program. Okay. And the flow layer contains a 4-channel imaging camera, a load input, discharge input, discharge outlet, and weather outlet. Under the control of microvalves in the control layer, the worms were sequentially loaded into the chamber, and live file image of their neural activities were automatically trapped. A fence-like structure was designed on gate 2 so that worms don't escape. And this part, in figure 3, the BLD model, which was developed for high performance live file recovery, is based on convolutional layers of subpixels. Convolutional layers, the convolution layers. There are 4 convolutional subpixel layers, and each one increases the image scale, and 5 convolutional layers were adopted for feature extraction, and 5 the convolution layers for image reconstruction. And this part, in figure 4, the sequential light file allow the recording of instantaneous position of C elegans in 3 dimensions, as well as the detection of a large number of worms of the high performance chip. The visual trajectories of of C. elegans wild type and UNC worms are different from worms wild type. In terms on their body curvatures, they are larger, have lower speed and shorter locomotion distance. And the conclusion, the microfluidic-based LFM approach is the first test to show high throughput handling of light samples in conjunction with 4D imaging of dynamic process, which can be beneficial on a car scale. Thanks.
Okay. I will talking about of this paper. High throughput tumor on a chip platform to study tumor stroma interactions and drug pharmacokinetics. Currently, available models of drug screening in oncology are not representative of the complex tumor microenvironment, resulting in the development of low throughput drugs. The L-tumor chip system is presented in this study. It's a three-layer microfluidic platform that integrates the microvasculature and environment of the tumor stroma in which normal fibroblasts, mesenchymal stem cells, and mm, cancer-associated fibroblasts, or CAF, were tested on cancer cell growth as well as pharmacological response to doxorubicin treatment. Okay. The chip consists of a top channel lined with a confluent layer of human endothelial cells and bottom chamber of matrigels with cancer cells separated um, by a middle layer with pores. Here you can see the flow pattern of the middle layer that allows controlled cellular communications and nutrient waste exchange between the bottom chamber and the top channel. The L-tumor chip chambers were seeded with the same density of breast cancer cells expressing red fluorescent protein, and the correlations between the intensity of the RFP signal and the number of cells was confirmed. The tumor chip system was composed of monolayer of microvascular endothelial cells, and to characterize the structure of the vasculature of this endothelial monolayer, first stained with two binding proteins, CD31 and BE cadherin. In this figure reflect the integrity of the immature endothelial banding of the tumor microvasculature. In addition, the permeability of the endothelium was measured by detecting fluorescent solutes such as 10 and 70 kilodalton dextranes, presenting greater permeability compared to normal endothelium. To study cell migrations, RFP labeled cancer cells were seeded in the bottom chamber, and endothelial cells were stained in the top channel. This figure shows the cancer cells crossed the endothelium into the top channel, while endothelial cells migrated, forming a spheroidal self-assembly with cancer cells in the bottom chamber. Cancer cells invasively disrupted the endothelium, gradually migrating to the top channel, highlighting their ability to directly alter endothelial permeability while at the same time forming more compact spheroidal structures within their original seeded chambers. To replicate the tumor microenvironment, normal fibroblasts or CAFs were introduced into the bottom chambers and co-cultured with tumor cells. Mesenchymal cells differentiated into CAF were confirmed by their elevated biomarker expressions. In this figure, normal fibroblasts reduce cancer cells growth, while cancer-associated fibroblasts and mesenchymal cells promote cancer cells proliferation. The study of doxorubicin, which is an anti-cancer drug, was carried out using a fluorogenic caspase assay. As shown in this figure, doxorubicin treatment induced cell apoptosis, as higher caspase-3 activities were observed in comparison between the treatment and untreated groups. The endothelium was also found to hinder the apoptotic effects of doxorubicin. In conclusion, the L-tumor chip system allows drug screening with a stage closer to in vivo conditions. Therefore, this potential use may facilitate the development of new anti-cancer drugs. That is all. Thank you.
Good morning, everyone. I'm going to talk about this paper called Microdroplet Enabled Cultivation of Single Yeast Cell Correlates with Bulk Growth and Reveals Subpopulation Phenomena. So here in the graphical abstract, we can see the process. So the aim of this study is to compare the behavior of yeast in single cell using microdroplets, and also compare it with the bulk culture, which is used by the industries in order to take advantage of the products that yeast produce. So here, these authors use microfluidic devices to generate droplets. As you can see here in the preparation, they use this microfluidic device that encapsulates yeast in microdroplets, and they monitor them and they examine them microscopically. So you can see at these time points how one yeast is encapsulated in this microdroplet, and after 2 hours it divided, then after 6 hours, 18 hours, 24 hours. And they also used GFP tagged yeast in order to capture images with the fluorescence, and to be able to count them as they form clusters, as you can see here at the 18 hours. And it's easier to count cells with images in fluorescence. All right, so let's divide the screen so that we can see the results. Okay, split, yeah. All right, so here they use, as Luis mentioned, this system also, the behavior of the yeast growth is also it's also fitted to a nonlinear regression to a sigmoidal logistic function. Here you can see the equation, and let's look for the graph. All right. So sorry, I think it's easier without the split. Just go down to the results. Yeah. So here we can see. So first, the yeast were plotted the number of cells per droplet is plotted in a logarithm scale. So you can see that the coefficient number is very close to 1 with this model, with the logarithmic scale. And you can see how the cell number per droplet increases and according to the time. Then here in this paper, they evaluated the stress when the yeast are exposed to acids as propionic acid. So here you see the different concentrations and how the number of cells per droplet changes when they are exposed to this stress. Also to the acetic acid, here you see how it changed. So the blue one is with no acetic acid, then the red one with this concentration, and the green one with the highest concentration. Here we also see in this figure how the yeast, the number of cells per droplet starts increasing according to the time, and they concluded that here there are some yeasts that have mutations, and also these mutations allow them to tolerate the concentration of propionic acid. So with microdroplets, they could study, and after destabilizing the droplet with this pico-break reagent, they could recover the yeast and then found the ones that tolerate to the propionic acid. Okay, so here you see the different strains of yeast that they used. And
1 minute left.
Thank you, Andrea. And finally, I wanted also to show you here that this is the design that they used, and this is how it looks with them under the microscope. And they use different cell concentrations in order to choose the correct one to have the majority of cells with just one yeast per droplet. So they choose the 3 10 to the 6 cells per milliliter, and that is, according to the Poisson distribution, the one that will have the highest probability to produce just one yeast per droplet. Thank you very much.
Hi everyone. I'm going to be presenting this paper, Coordination Polymer Nanoglue, as a robust addition based on collective lamellar stacking of nanoplates. Coordination polymer is an inorganic or organometallic polymer structure containing metal cation centers and linked by ligands. The collective properties of the coordination polymer colloids have rarely been addressed, mainly due to the difficulty in assembling them into superstructure with impressive mechanical strength. So here, the authors demonstrated that coordination polymer nanoplates could assemble together spontaneously by a liquid evaporation-driven stacking method, and that the stacked coordination polymer nanoplates could work like a polymeric adhesive. So the nanoplates are approximately 180 nanometers by 14 nanometers. So when a drop of the nanoplate suspension is sandwiched between 2 pieces of slice, and after drying them in open air to remove interparticle water, the 2 pieces of slides are glued together. This bond is quite stable. For example, a heavy load of 2 kilos can be held in a small place area without causing the attachment. The addition effects of the nanoglue was investigated. A force displacement curve shows a slip-stick structure, suggesting that the nanoplates slide at first and then interlock with each other gradually. The addition strength is correlated to the loading density of the nanoplates, so the higher loading mass of the nanoplates per area, the stronger the adhesive strength can be. The shear addition strength of the glue glass slides are kept very well even after treatments such as heating up to 200 degrees under vacuum or cooling it in liquid nitrogen, and even thermal recycling. They also deattach the glue glass slides and recover the adhesive strength by wetting with water droplets. So this nanoglue, as you can see in E, combined a large variety of materials such as glass, ceramic, metals, woods, polymers. It doesn't matter if the surface is flat or rough. So this versatile binding result clearly suggests that the adhesion property is not specific or limited. And for instance, it can glue PTMS as well as macroporous materials. The nanoglue can not only work for macro-sized substrate, but also for mesoscale objects. For example, it can bind colloidal cubes to the surface of aluminum foil, and if the colloidal cubes are coated alone, they can be peeled off easily by folding, indicating that the robust coating of these cubes relies on the utilization of the nanoplates. To understand the origin of the adhesion effect, the role of the size and the shape of the nanoglue was investigated, and when they changed the lateral size of the nanoplates to 110 nanometers, significant shear adhesion strength was retained. However, when the lateral size is over 1 micrometer, the shear adhesion strength becomes very weak, suggesting that laminar stacking is necessary. They also changed the shape into irregular microparticles, and the adhesion effect got lost, suggesting that this adhesion effect probably comes from low dimension anisotropic shape of the nanoplates. Besides the ability of to glue other surfaces, the nanoplates can glue themselves as well. By evaporation of the water at room temperature, different nanoplate-based bulk structures can be fabricated. This bulk material is constituted of microdomains of stacks nanoplates and in different orientations, indicating that the sample is isotropic. The obtained bulk structures are stable at high temperatures and in vacuum, and after heating up to 100 degrees under vacuum, the shape of the bulk structure did not change, although a color change happened because of the removal of coordinated water. Also, the assembled back structure do not lose their absorption capability, and furthermore, the bulk structures keep their morphological stability even after absorption and desorption of gas molecules. Finally, regarding the nanoplates adhesion mechanism, although they are not flexible and interlock molecules change, like with polymeric adhesive, the multi-layer assembly of the nanoplates allow the mimicking of this polymeric adhesive. When the slurry of the nanoplates is coating on the substrate, the water allows a full wetting of the surface, and after drying up, the nanoplates organize in a lamellar way to form multi-layer assemblies. The van der Waals forces existing between the nanoplates cause a cohesion effect to render the slip between the two nanoplates, making the nanoplates integrate robustly. As the van der Waals forces universally exist among all the materials, the adhesion effect can happen between the nanoplates and other substrates like glass, aluminum, PET, and all the other mentions before. The different orientation of the microdomains drive the multi-layer assembly of the nanoplates to interlock with each other, fixing the substrates with the nanoplates. So the function of a polymeric adhesive can be successfully mimicked by the multi-layer assembly of these nanoplates. That is all. Thank you very much.